Hi, my name is Shally, and I am a fourth-year undergraduate student doing research under the supervision of Dr. Bob Goldberg in the Molecular Cell and Developmental Biology Department. And today I am excited to share with you the research that I am currently doing in determining the role of the WOX9 transcription factor in the heart stage of soybean seed development. And so first, I want to start off by explaining why we study soybeans in the first place. And the answer to that is food. Soybeans are one of the largest crops grown in the US and around the world. It is used for both human consumption as well as livestock feed and biofuels. And in 2022, we are projected to reach 87.5 million acres of soybean crop in the US alone. And to put that into perspective, that is roughly the size of 2 Floridas. So soybeans are an extremely important crop for both our economy and food sources. However, as climate conditions worsen in the coming decades, it will become harder to grow enough food to feed our growing human population, particularly in areas of the world that are currently responsible for a lot of our food production. And by 2050, we are projected to reach 9 billion people. So in the next few decades, we are going to need to produce more food than we ever have before in order to feed our growing population in worsening conditions. So by studying soybeans and other plants, we can figure out exactly how this plant grows and the genetic processes that control the development of these plants. And the goal is that eventually we can manipulate those genes to produce plants that can grow faster or have more nutrients or are drought resistant, and a number of other characteristics that are all going to be helpful in feeding the world. And understanding the soybean will also give us information on other legume crops and seed development in general, because it is an excellent model organism. And finally, soybeans are a great plant for studying genetics because its entire genome has already been sequenced. So looking at the development of a seed, these are the major stages. The first 3 stages, the globular, heart, and cotyledon stages, is where morphogenesis of the seed occurs. So the differentiation of all the different regions and tissues in the seed. The next 3 stages, early, mid, and late maturation, is when storage reserve deposition occurs. So lipids, oils, and proteins accumulate as the seed matures. And by the end of our maturation stages, we have a dormant seed that can remain in this dormant stage until the conditions are right for germination. And in particular, these early morphogenesis stages are of interest to us because this is where a lot of the really important decisions are being made about where all of the different tissues and subregions of the seed differentiate. And one of the earliest processes to occur right after fertilization is the differentiation of this structure called the suspensor. And the suspensor is a temporary structure that provides support to the growing embryo proper. And the embryo proper is the part of the embryo that becomes the actual new plant. So the suspensor attaches the embryo proper to the maternal environment. And so it's, in a sense, it is kind of like a plant umbilical cord. So obviously this is a really important structure and step in development. So a major question we want to answer here is, what are the regulatory networks controlling gene activity in the embryo proper and suspensor? So in other words, how does the embryo know to divide in this way? What are the genes that are controlling this process, and what is regulating those genes? And so this is where transcription factors come into play. Transcription factors, in case you're not familiar with cell biology, are the proteins that regulate gene expression. So essentially, they act as on and off switches for genes. And through transcription factors, different genes can be turned on and off in different tissues and different stages of development. And so to find out what transcription factors are responsible for regulating gene expression in the suspensor, previous work in our lab studied the genes expressed in 4 different plants: the scarlet runner bean, common bean, soybean, and Arabidopsis. And this work found 6 different transcription factors to be expressed in the suspensors of all 4 of these plant embryos. And especially interesting because these plants have very different morphologies. And in particular, WOX9 was prioritized because it was the most prevalent, and its role was further studied in the globular stage, which is the first stage of development. And WOX9 is a homeobox gene, which are genes that regulate embryonic patterning in most organisms. And this research found that WOX9 is involved in regulatory networks. It regulates genes that are mostly involved in the regulation of transcription. And interestingly, 2 of these target genes are actually in the original set of 6 that we found in all 4 of these plants. So this tells us that WOX9 regulates other genes that also regulate genes and form this network of genes that interact with each other to control suspensor differentiation. And all of this work was done in the globular stage. And so this brings us to the major question of my research, which is what are the WOX9 target genes in heart stage soybean seeds? So the very next stage following the globular stage. And since by the heart stage, the suspensor tissue has already been differentiated, my hypothesis is that WOX9 is still involved in a regulatory network during this stage, but it is now controlling the further development and function of the suspensor rather than its differentiation. And so to do this, I performed chromatin immunoprecipitation on heart stage soybean seeds, which I will explain in further detail later, and analyzed it using Qpcr and sequencing. And so the first step, obviously, is to collect heart stage seeds. And so here we have a diagram of the soybean seed at the heart stage. And as you can see, it's called the heart stage because the embryo is shaped like a heart. And at this stage, we know that the size of the seed is 1.7 to 2.1 millimeters, and the size of the pod, which typically contains about 3 seeds, the pod is 1.5 to 2 centimeters. So I collected soybean seeds based on these size requirements. Then the next part is chromatin immunoprecipitation, or CHIP for short. And this experiment allows us to determine the particular sequences, the genes that a protein of interest is bound to. So in this case, WOX9. And the first step in this CHIP procedure is to cross-link proteins to the DNA. So what this means is essentially we are locking the proteins down onto the DNA exactly where they are bound at this moment. And then we can isolate the DNA and break it up into a bunch of little fragments. And after that, we use antibodies to pull down just the fragments of DNA that are bound to WOX9. And then we would purify the DNA. And with that DNA, we can do 2 things, sequencing and Qpcr. And so sequencing allows us to find the exact DNA sequences the WOX9 was bound to. Then we can match those sequences up to our already existing soybean genome to find out exactly what those genes are. And then once we know what the target genes are, Qpcr, which stands for quantitative polymerase chain reaction, is a way of measuring the amount of DNA in a sample. And so that lets us know how much WOX9 binding we have to these target genes. So in summary, my project is on elucidating the role of WOX9 in the early stages of soybean seed development. And in previous experiments revealed that during the globular stage, WOX9 regulates genes that are involved in the regulation of transcription. And so my current project is to determine the role of WOX9 in heart stage soybean seeds. And to do so, I conducted chromatin immunoprecipitation experiments, and I'm currently waiting to sequence those samples. And so with that, that is the end of my brief talk. And I want to thank my lab, the Goldberg Lab, for allowing me to do this research, as well as the integrated and interdisciplinary undergraduate research program faculty and students for helping me develop my skills as a researcher. And if you have any questions or would like to know any further details about my research, please feel free to contact me at my email here. And thank you so much for your attention.